In a major scientific advancement, artificial embryos modelled with stem cells have emerged as a way of studying early development. However, as a recent report from the UK's Parliamentary Office of Science and Technology has highlighted, they're currently unregulated, and so ethical decisions need to be made soon about how we can make use of them. So in this video we're breaking down that report to understand what artificial embryos are, what their research potential is, and why their ethics are proving tricky to regulate. Within the world of study of human embryo development, there has been a major scientific advancement made. That is the ability to generate embryo models out of human stem cells. Stem cells are the type of cells which are almost like a raw material. They have the unique ability to develop into all other types of cells in the body. Now, when it comes to these embryo models that have been developed, the products of them are called stem cell-based embryo models, SCBEMs. For the rest of this video though, we'll call them stembryos. They actually have lots of different names, which we'll get onto a little bit later, but anyone who knows me knows I like a pun, so stembryos it is. Anyways, a little bit about them. Stembryos come in a variety of different models, ranging in complexity and how wholly they model an embryo. Stembryo models form a wide spectrum, but they can be classified into two types: non-integrated and integrated. Non-integrated stembryos are used to model a specific part or developmental process of a developing embryo, and importantly, don't include extra embryonic cells. These are the sorts of cells that develop into things like a placenta or an umbilical cord, which help the embryo develop into a fetus but aren't actually part of the thing itself. Because non-integrated stembryos lack these, they lack the ability to develop into a fetus. However, integrated stembryos do develop with extra embryonic tissue. The quality of the models isn't amazing right now, but it's thought that as they improve in the future, all of the component pieces are there so that integrated stembryos could acquire the potential to develop into a fetus. If you think that sounds like it could be an issue, then you'd be right. But we'll get onto that in a moment, because first we have to ask that given we have all these stembryo models, then what can we actually use them for? Well, they allow for new forms of study into embryonic processes, means of study that wouldn't be possible or at least practical with human embryos. Therefore, they represent a scientific advance and an ethical advance towards a better understanding of embryo development. Practically, that understanding could extend to a few different topics. When it comes to IVF treatments, right now about 50% of fertilized eggs fail to develop, but some scientists think that study of stembryos could help reduce this failure rate. Their study could help with natural conception too, helping to identify and reduce risks of early pregnancy loss, an issue which affects approximately 1 in 5 women in the UK. The research could also investigate various birth defects, ranging from malformations in the spine to congenital heart disease, one of the leading causes of death in newborns. Toxicity studies of chemicals that harm growing embryos could also be carried out. They're done so on animals like mice already, but this doesn't account for species-specific responses. For instance, when testing thalidomide, a morning sickness drug which results in severe birth defects in humans, it has a greater impact on human stembryos than on mouse ones. Responses like that, where the response is stronger in humans, could be identified with this sort of testing. So, stembryos are a major advancement with a lot of research potential, but being something new, they are not yet explicitly regulated in the UK, and that's a problem. Traditionally, public bodies oversee research and treatment using human embryos and some categories of stem cells to ensure that there is a strong ethical and social foundation to their use. The combination though, stembryos are at this point unregulated, and so whilst that's the case, there's a risk that they'll be developed irresponsibly or immorally, causing human indignities and going against societal values. The trouble is, just as they're a new technology, they've brought new challenges that need to be overcome so that we can properly regulate it. First of all, and this is a bigger problem than you might think, nobody can decide what to call them. Across scientific, ethical, and legal literature, a whole range of different terms has been used to describe them. Artificial embryos, synthetic embryos, stembryos, synthetic human entities with embryo-like features, embryo-like structures, embryo models, embryoids. Each of these can have different impacts on public perception, lead to misinformation, and generally cause a headache for regulators. And despite ongoing efforts to decide on a preferred term, it's actually unclear still what the consensus on this will be. Look, if we're taking a vote on it, I like stembrio, I like the pun. Names aside though, there is a big discussion over the similarities and distinctions between a stembrio and an embryo. As the technology of stembryos develops, there may come a time in future when these models, these artificial stembryos, are indistinguishable from real human embryos. What would make that so? Well, the stembryo's ability to develop into a fetus. But there's a catch. The only way to completely test this would be to transfer a stembryo into a human host. And that is a procedure which is not legally permitted in the UK. Now, a similar test could be carried out on animals instead, or alternatively you could see if a modelled stembryo can develop for a long time and accurately enough in vitro, in glass. But that duration has to be an ethically acceptable one, which brings us on to a whole other issue: ethical time limits on stembryos. Study of embryos can be regulated fairly easily, as their development follows a clear path over the course of days and weeks. So in the UK they can be maintained ethically up to 14 days, in accordance with the Human Fertilisation and Embryology Act. Stembryos, however, don't have an equivalent phase of fertilisation, and so there is no day zero to start counting from. Add to this the fact that models develop at different rates, and that this development isn't linear, perhaps skipping or showing different sequences of development. Setting time limits on their usage is quite tricky. Some suggest that the solution could be to not model in time, but to model instead in the development and emergence of certain morphological features, like the primitive streak, evidence that a body plan is beginning to form in an embryo around 14 days after fertilisation. Others argue that there shouldn't actually be any need to develop stembryos any further than this at all, citing that there are other methods of study for investigating organs and tissue development in isolation. Whatever case is made though, it needs to be written up in regulation. Few countries are ahead of the curve and have started to do this, defining stembryos versus embryos and setting time limits. But they're not all in agreement. In Australia, stembryos fall within the definition of a human embryo, and so the regulations are carried over, restricting maintenance for study to up to 14 days or the equivalent morphological development. In Austria, there's no explicit regulation, but the Ethics Academy of the Austrian Academy of Science delivers licenses for study on a case-by-case basis. And in France, stembryos have been proposed to be different to embryos on the basis of their origin and their intentionality, and it has been proposed that a 28-day maintenance restriction be put in place. What are the leading groups in the sector concluding though? Well, for that we can look to the International Society for Stem Cell Research, the ISSCR, an international scientific society made up of scientists, educators, ethicists, and business leaders. In 2021, they updated their guidelines to classify stembryos into the non-integrated and integrated classes we discussed earlier, subject non-integrated stembryos to an oversight process, and integrated stembryos to additional review, one that requires compelling scientific rationale to work with and the minimum maintenance time necessary. They now prohibit the transfer of a stembryo into the uterus of a human or animal, and they removed their prohibition of maintaining human embryos past 14 days, pending a reassessment with public communication. The note of public communication is a big one. Public engagement is central to the governance of stembryos. Transparency, consent, and ethics have emerged as key requirements emerging out of various communications and workshops between public and professionals around the topic. Plus, as mentioned earlier, it's important to have a clear and grounded scientific narrative to avoid misinformation. Today, though, in the UK at least, the topic of stembryos remains up in the air. In November 2023, the UK's Human Fertilisation and Embryology Authority delivered a report to the UK government with various recommendations about how to approach regulating stembryos. But as well as that, there were also some broader recommendations about future-proofing scientific developments, perhaps by introducing a trial mechanism for various new products to be trialled in a controlled manner that would regularly report back to Parliament. Right now, the UK government is still considering that report and the reforms that it laid out, and is set to respond in due course. Hopefully, though, you watching have learnt something, and now will know more about the science informing that future policy when it comes. That's all for this episode of Science Affairs. If you enjoyed it, then be sure to like the video and subscribe, and don't forget to ring the bell to be notified of when the next video is released. Thanks for watching.